When studying any of the sensory modalities, such as hearing, vision, taste, olfaction, and so on, a good approach is to ask some basic questions. And these are, in fact, commonalities among the different sensory modalities. What characteristics of the physical stimulus can we actually detect? Where and how is energy transduced into neural impulses? Does adaptation occur with prolonged stimulation? And how are the properties of the physical stimulus encoded in the nervous system? Which parts of the brain are particularly important for detecting and analyzing sensory input? And how do we integrate information from more than one sensory modality? I'll be using vision, human vision specifically, as an example of how to approach these questions. The first question to ask about any sensory modality is, what is the nature of the physical stimulus? We're going to use vision, the human visual system, as a model for how to study information about the different sensory modalities. I'm not sure why, but there have been quite a few students over the years who said this is their favorite part of the entire general psychology course. So we'll see what you think. Looking at the guiding questions in your study aids, the first question is again, what is the nature of the physical stimulus that's being detected? And we're talking about vision, so what is that physical stimulus? It is, of course, light. And what is light? Light is part of the entire spectrum of electromagnetic energy that surrounds us. It is composed of electrically charged matter that oscillates as it moves through space. What we detect as light is really only a tiny portion of this entire electromagnetic spectrum that ranges from radio waves to cosmic waves. Looking at the diagram on the bottom left of the screen, I've drawn a waveform. We call it a sine wave. It goes up and down and up and down. And we can speak of the amplitude of the sine wave or its height. And with light, amplitude corresponds to the intensity or brightness of the light. A sine wave can also be described as having a wavelength, and that's represented by that lambda above the bidirectional arrow. And the wavelength really just refers to the distance between two adjacent peaks of the sine wave. With vision, of course, the wavelength of light corresponds to the color that we perceive the light to be. As we consider the electromagnetic spectrum that surrounds us in our environment, ranging from radio waves to cosmic waves, you can see that radio waves have a wavelength of 10 to the 6 meters. That is huge. The distance between the peaks of the waves is 10 to the 6 meters. That's a 1 and 6 zeros, or a million meters, and a meter is about 39 1/3 inches. So can our body detect radio waves? No. We have to build instruments in order to detect the radio waves in the environment around us. At the other end of the spectrum, you have cosmic waves. Here, the distance between adjacent peaks of the sine wave is 10 to the minus 14 meters, which is tiny, tiny, tiny. Can our bodies detect cosmic waves? No. We need to build instruments in order to detect their presence. But looking in the middle of this electromagnetic spectrum, you will see the tiny portion of it that we can detect with our bodies and that we experience as light. You'll also see that I made an error drawing this diagram. To be more accurate, the sine wave would have to go up and down many more times between the 800 nanometer and the 400 nanometer portion of it that we detect as light of varying wavelengths. But it's still a useful diagram and the best that I could do. Note that the portion of the electromagnetic spectrum that we can detect with our eyes and that we experience as light ranges from about 400 to 800 nanometers, or light that we detect as bluish up to light that we detect as reddish. On the bottom right of the screen, I've shown you that 1 nanometer equals 10 to the minus 9 meters, or a billionth of a meter. And again, a meter is about 39 1/3 inches. I am not going to test you on those values, but I think it's helpful for you as you try to visualize the portion of the electromagnetic spectrum that we perceive as light. And this answers the first question in the study aids when we're thinking about vision. What is the nature of the physical stimulus? And this is how you would describe it. We also need to consider the structures in the body that allow us to detect light. This begins with a discussion of our eyes, and I've drawn an eyeball here, and we need to talk about the parts of the eye. So this eyeball is looking to the left at a flower, and as light is reflected from the flower and enters the eye, it passes through the cornea at the front of the eye, which is a transparent outer covering. The majority of the eyeball surface is a white tough substance called the sclera. As light enters the eye, it passes through the cornea, through the pupil of the eye, and the pupil is the opening in the center of your iris, and the iris is the part of the eye that gives your eyes their color. Some of you have brown eyes, some blue, some green eyes, and I once saw a student who had purple eyes, which was gorgeous, but that's irrelevant here. At any rate, light enters the eye through the cornea, it passes through the pupil, and the pupil is of interest to psychologists for a variety of reasons. One is that it can, at times, indicate our emotional states. In fact, the pupillary response is something that's often measured by psychologists. You probably already know that the size of the pupil changes as a function of the amount of light in the room, the ambient light. It is constricted when the light is bright, or it is wider when the light is dim. And this protects the structures of the eye from being exposed to too much light. But the pupillary response also can show our emotional states. This is a bit pop psychology or pseudoscience, but if you're talking to someone and the room lighting is stable, and their eyes seem widely dilated, and you happen to know they're not on any kind of psychoactive substances, then perhaps you can assume that that individual is sort of in the moment with you, listening to you closely, and you have a hint at least about their emotional reactions to what you're saying. Going back to the structures of the eye, once light enters through the pupil, it enters the front chamber of the eye that contains the aqueous humor, and then it passes through the lens of the eye. Now, the lens of the human eye is convex in shape, meaning it's thicker in the middle and thinner at the edges. But it is also held in place by a series of muscles that will stretch out the lens or allow it to relax, thus changing its shape. This process is referred to as accommodation, and it allows us to see objects at varying distances. I can stand in the classroom and see students in the very back of the room, or I can see the students in the very front of the room. The lens of my eye changes shape, and I can focus at a distance or focus nearby. After light passes through the lens, it enters the back chamber of the eye that contains the vitreous humor. The vitreous humor is a gel-like substance that gives the eyeball its shape. After light passes through the back chamber of the eye, it reaches the retina. And this will be the primary focus when we talk about vision, because this is where light, environmental energy, is changed into neural impulse. But there are other important structures to discuss first. First of all, I want you to note that at the center back of the eyeball, there's a structure that I've identified as the fovea. And this is a part of the eye that is important for high acuity or highly detailed vision. The fovea is located at the back center of the human eye. And if you think about it, if you want to see details of an object, do you look at it out of the corner of your eye, or do you look at it straight on? If you want to see the details, you look at an object straight on. That image is projected in the back center of the retina, and that's the fovea, and that's the location of the retina that's important for high acuity or highly detailed vision. Televisions have changed, but it used to be that you could sit just a foot or so from a television set, and you would see the little dots, the little colored dots that gave rise to the television image. When you did that, you could close one eye, stare straight ahead at the television image, and you would see all those dots. But the dots that you would see were just in a circle in the center of your vision. In the more peripheral parts of your visual field, around the edges, the dots would all blend together, producing the normal TV image. I always thought this was really cool because you could see the size and shape of the fovea, a circle in the back center of the retina. I'm still looking for a similar situation for students to be able to have this experience. Let me know if you can think of one. There's another structure that I want you to know. It's the optic disc, and this is where the optic nerve leaves the eye. All the information picked up by the retina leaves the eye via the optic nerve, and the optic nerve gives rise to a blind spot in our visual field. More detail than you actually have to know, it's the axons of ganglion cells that leaves the eye as the optic nerve. If you've got axons of nerve cells there, you cannot have the photoreceptors. So you cannot have two things in the same place. So the optic disc is associated with a blind spot in the visual field of each eye. And you can try this. The optic disc is located nasally for each eye. So you can try closing one eye, stare straight ahead, and hold up your forefinger in the periphery of your visual field. Move it around until your finger disappears. When the image of your finger is projected onto the part of the retina that is the optic disc, there are no photoreceptors there, and so the tip of your finger literally disappears. Personally, I've always had difficulty doing this. It might work for you, but it did happen to me once in class. I got very excited. It happened to me once in my whole life. It often works for students, on the other hand, and if you try this, it'll help you remember that the optic disc gives rise to a blind spot in a visual field. Now it's time to talk a little bit more about what happens to light as it passes through the human eye. You may already be aware that the lens of the eye is important for refraction of light, bending of light rays in order to focus an image clearly on the fovea at the back of the eye. To help you understand this, I want you to imagine standing outside on a sidewalk on a hot day. Sun is out and it's bright and shining, and you're holding a magnifying glass. And you hold that magnifying glass so that the image of the sun is projected onto the sidewalk. You can move the magnifying glass up and down, and as you do so, the image will be fuzzier looking, or you can move it and find the distance above the sidewalk where the image of the sun on the sidewalk is crisp and clear, and all those light rays are being bent, refracted, focused into a clear image of the sun on the sidewalk. When you do that, if you find that point, you could probably place some dry leaves in that spot and they will burn. Now think about the lens of the eye. It cannot move back and forth in order to focus images on the retina clearly at the back of the eye. Instead, you know that it undergoes accommodation because there are muscles holding it in place, allowing the lens to change its shape. What you may not know about the human eye, though, is that the cornea of the eye also refracts light, and in fact it provides about 70% of the eye's refractory power, bending light, focusing images clearly on the retina at the back of the eye. The lens provides less of the eye's refractory power, but it can undergo accommodation, or changing its shape, in order to see objects at a distance or close up. One more thing about the lens of the human eye: because it's a convex lens, an image is projected upside down. So you see an eyeball looking at a flower in this diagram, and the image of the flower is being projected upside down on the retina. That's just the nature of convex lenses. But you would perceive the flower as right side up. If the flower were presented to the eye upside down, it would appear right side up on the retina, and you would perceive it as upside down. Now I'm going to introduce you to what may be the most important vocabulary term of all when we talk about human vision, and that vocabulary term is transduction. Transduction refers to the conversion of environmental energy into neural impulses. And if you think about it, this is incredibly important. How is it that energy outside of your body in the environment changes into neural impulses that are part of your perceptual experiences? In the human eye, transduction takes place at the retina, where there are photoreceptors, specifically the rods and the cones. I've drawn a magnified view of the retina at the back of the eye, and it shows the rods and the cones. The rods are literally rod-shaped structures, and I've shown them here in red. They're not really red. And the cones are literally cone-shaped structures, and I've shown them here in blue. And you know they're not actually blue as they appear in the human eye. When you look at this diagram, you'll see that the rods and cones are connected to other cells. These cells include ganglion cells, amacrine cells, bipolar cells. I'm not going to ask you to memorize that information, but you should know that there are neurons in the retina that receive information from the rods and the cones, and some visual processing does take place at the level of the retina. Then that information travels along the optic nerve, leaves the eye at the optic disc, and eventually that information goes to the thalamus and the occipital lobe of the cerebral cortex, as well as other parts of the brain. When we talk about how transduction takes place at the retina, then we are essentially discussing the functions of the photoreceptors. Photo means light, a receptor receives something. So those photoreceptors in the retina are the rods and the cones. Let's take the rods first. How do they do this? How do they transduce light into neural impulses? It's actually a chemical process. The rods contain a substance called rhodopsin, which is also called visual purple. Rhodopsin can be removed from the rods, placed in a test tube, and it is a purplish color. If it is exposed to light of a particular wavelength, then it undergoes a chemical process called bleaching, and it turns kind of a whitish pink color. You do not need to know this, but rhodopsin undergoes its chemical change optimally when exposed to light of about 505 nanometers. But what's important for you here is that the rods contain a substance called rhodopsin. It undergoes a chemical change when exposed to light of a particular wavelength, and that is the basis for transduction, or the conversion of light energy in the environment into neural impulses in our nervous system. Now let's talk about the cones of the eye. You probably already know that the cones of the retina are important for our color vision. And for most people, there are 3 different types of photopigments, not just one like the rods. In the cones, there are 3 types of photopigments that are important for transduction of light. There are photopigments in some cones that are optimally sensitive to light of about 700 nanometers, long wavelength light that we perceive as red. There are also some cones that have photopigments optimally sensitive to light of about 540 nanometers, or medium wavelength light that we perceive as green. And finally, there's a 3rd type of photopigment that undergoes a chemical change optimally or best at about 480 nanometers, or light that we perceive as blue. Each cone cell in our retina contains just one of these types of photopigments, and these various photopigments give rise to our entire experience of color as we perceive the world around us. I'm not going to ask that you memorize 700 nanometers, 540 nanometers, or 480 nanometers, but I do want you to know that these are often considered to be the 3 primary colors. Red is long wavelength, green is medium wavelength, and blue is short wavelength light. I should also mention that the fovea of the eye is rich in cones, and so the fovea is not just important for high acuity vision, highly detailed vision, but is also important for color vision. And one way that you can test this is ask a family member or a friend to sit in a chair and face forward, stare straight ahead, and you stand behind them with a pen or some sort of object, say it's a red object, and you gradually move that object around their head until they say they see it. As they stare ahead, the object is projected onto the periphery of their retina, and they will not see the color, but they'll detect that there's something there. Keep moving that object around their head until it is in front of their face, and there will come a point where they can say the color of the object that you're holding, and that is when the object is almost in front of the face, held so that the image of the object is projected on the center back of the retina, or the fovea. I said that the fovea, or the center of the human retina, is rich in cones. As you move away from the center of the retina, you encounter more and more rod cells. And obviously the rods are not important for color vision because there's only one type of photopigment in the rods. But here's what the rods can do. The rods are important for vision in dim light. When objects are projected on the periphery of the retina, we may not see color or we may not see details, but we can be very sensitive to the presence of that object. Try this out. Go outside on a starry night and look up at the stars, and as you stare straight up, you may see a very dim star twinkling in your peripheral vision. When you turn to look at that dim star directly, it disappears because now the image is projected onto the fovea, which is not that sensitive to dim light. Turn your head again so that you are looking at that dim twinkling little star out of the corner of your eye, so that it is project it onto the periphery of your retina where there are many rod cells, and you'll see it again. So now you know a little something about the neuroanatomy of the retina. You have the fovea rich in cones at the back center of the retina, important for high acuity vision, and then as you move away from the center of the retina to the more peripheral parts, you have more and more rod cells, and they're important for vision in dim light.